This conference will now be recorded.
All right, folks, so good morning and thank you for attending today. Today we're going to be finishing up the chapter. Now, I haven't done a full official lecture on chapter 5, but I'm going over all the images for the different types of tissues, and I'm going to finish that up. We already had reviewed epithelial tissue, and we know that there are, whether we have a simple one-layer group of cells in the tissue or stratified multiple layers of these cells, and whether they're squamous, whether they're columnar or cuboidal, right? We've learned all that information, and so you will need to, I need to shut off my email here. Let's do that. So that's good, okay. So you will need to review the different types of tissue. Remember that epithelial tissue covers and lines. It's avascular, right? There's no blood vessels present. And differentiating pseudo and regular. Yeah, so pseudo, right? So as we're looking at pseudo-stratified tissue, what you're going to see, folks, is that as you're looking at this type of tissue, so you're going to notice that it's going to look like there are multiple layers here, right? But you're going to see that at the apical surface, we have the cilia present, and there's also quite a bit of cytoplasm present, okay? So this helps you to have an understanding of, hey, as I'm looking at this type of tissue, really, when we look at transitional epithelial tissue, or when we look at stratified squamous epithelial tissue, now if it's non-keratinized, you're going to see that at the apical surface, you're going to see nuclei, okay? And when you're looking at stratified squamous that's keratinized, this is a very good example. You're seeing here that, look at this top layer of this cell. So here we have multiple layers, but we only address the type of tissue, identify the type of tissue by looking at the apical surface. So here we have this apical surface, and these are all dead cells, right? The epidermis, right? So non-keratinized, look at the apical surface. So we're seeing that we have nuclei present here, and they're flat cells. So that would be in the oral cavity, in the vagina. These can be areas where non-keratinized stratified squamous epithelial tissue. So it's much different than when we're looking at pseudo-stratified. It looks like this, but we can see at the apical surface, here's a lot of cytoplasm, and here we have these cilia, which are commonly present there. So that's when you're noticing and looking at pseudo-stratified, that you just have to look at multiple images, folks. It takes a long time to understand that, and that's why, sure, Will, you're welcome. And you really have to just review many images over a period of time. Recall that histology-world.com can also be helpful. And I'm telling you, just doing this as far as Google images of the types of tissues will absolutely help you. So we're going to be looking at areolar connective tissue first. This is also known as loose connective tissue. So let's take a look at this image right here, and this is a very good image. So what we're seeing here with this image is that you're seeing these elastic fibers that look like almost like someone took like a pen or a marker and just painted these dark lines. And then these thicker lines, these are collagen fibers, elastic fibers. And then you have many of these cells would be considered fibroblasts. These are fiber synthesizing or fiber producing cells. Areolar connective tissue, it's a packing type of tissue present throughout the body and also present within the subcutaneous layer of the skin, the supportive layer of the dermis of the skin. Areolar connective tissue. Now, that's also, like I said, considered a loose connective tissue. A dense, regular connective tissue would be an example right here. This is a very good example of And what we're seeing here, folks, is that we have this dense regular connective tissue, and let's make it bigger. And what you're seeing here is that there's a wavy pattern to much of this type of tissue. These pink lines represent collagen fibers, collagen fibers. So think ligaments, think tendons. A tendon will connect a muscle to the bone. A ligament will connect bone to bone, right? They're connective type tissues, right? As the areolar connective tissue, dense regular connective tissue, they'll connect the tissues, okay? Very important. And when you're looking at it, you'll see that, yes, there are multiple nuclei as well as these wavy collagen fibers, dense regular connective tissue. Now, as we look at adipose tissue, in adipose tissue, you'll see here that they look like soap bubbles. Adipose connective tissue, this is fat, right? Adipose tissue. These cells are adipocytes. They are fat cells, okay? You can, I just, when I look at them, I see these irregular shaped, oval shaped cells that are really, it's a full cell, right? You see here, that's a fat cell, right? Adipocytes, adipose tissue, okay? Look like soap bubbles. Different from, I've shown you as far as the lung tissue histology, where I've shown you simple squamous epithelial tissue, where it's one. So look at this for a moment, and it looks similar, but it's different, folks. Okay, see this alveolus? These are alveoli, these little sacs present within the lungs, where we have gas exchange occurring. But you'll see here, folks, that they're not all, so like that's together, but this is not. And all of these little areas here, so let's get a, when you're seeing these lines of tissue, these are cells, and these are simple squamous epithelial tissue. But you see how it's different from, similar looking, but different from simple squamous epithelial tissue. Because what you're seeing here, folks, is that there are these little simple squamous cells creating these borders present where there's gas exchange occurring. So let's look at this. No, go back. Here's another one. Oh, that one. There we go. Again, you're seeing here that these are simple squamous, similar to, but you have to look at many images to see that does look different than this is the adipose tissue, simple squamous epithelial tissue, okay? Then we get to reticular tissue. This reticular tissue is quite interesting, and I'll find, now this is not going to be in the lab. We do not have slides of the reticular tissue, but what I would like you to see would be that you'll see that reticular tissue is present, and I just show it to you. That's just how you get an idea. Reticular tissue is present within lymphatic tissue. This would be a part of your immune system and your lymphatic system, right? Your immune response. It kind of looks like, and I tell students, and I've heard this from other professors making mention of this, and it makes sense, that the dark lines represent, it looks kind of like grapes on a grapevine. Take a grapes on a grapevine. That's what the cells kind of look like. They're reticular connective tissue. It's not going to be in the lab. You're not going to have to identify this. We don't have slides for reticular connective tissue, but just showing you what this looks like. It's present within the lymphatic system. Let me show you on your PowerPoint here, and you'll go. It's taking me a moment. Sorry about that. Let's see. Dense regular. There you go. Reticular. So you see here, this is slide number 28 in the PowerPoint. forms a framework of lymphatic tissue, hemopoietic tissues, and spleen. The spleen is a part of the lymphatic system. So say like the lymph nodes and such, this is all reticular tissue. Hemopoietic tissue is just tissue that produces bone cells, okay? You'll see network of fine reticular fibers. These reticular fibers, so the vines for the grapes, right, that you're seeing on that image there, the reticular fibers are fine collagen fibers. That's what they are. They're very thin collagen fibers. So reticular connective tissue. Now, next we're going to talk about another type of connective tissue, which would be a type of connective tissue that is cartilage, okay? Now I'm going to show you that there are 3 different types of cartilage: elastic, fibro, and hyaline cartilage. I'm going to show you for elastic and fibro cartilage how to identify what they look like and where they're located, and then every place else in the body would have contained hyaline cartilage, okay? So when we're thinking of elastic cartilage, this is really what we're looking at. This is elastic cartilage, this region right here. You'll see that the dark fibers of the elastic elastic fibers, these dark lines here of the elastic cartilage. You have these chondrocytes, these cartilage cells present within a space. This is called a lacuna, or lacunae plural. Look at this image here. You'll see elastic cartilage. You'll have here these chondrocytes present within a space called the lacunae, and you have these fibers, these elastic fibers, and they look just like this, folks. So it's very easy to represent and to see this and go, oh, hey, that's elastic cartilage. Very easy to identify. Fibrocartilage really is going to look really in the lab that we have the examples of. It's going to be stained blue, and you're going to see here, let's show you this for a moment. Here, they also has these fibers present, these collagen fibers present, and it has these cartilage cells, these chondrocytes present. And you can see them, the dots, those are the cartilage cells present in lacunae, and these are the collagen fibers of fibrocartilage. Now, let me step back for a moment here and say that elastic cartilage, think the E's. External ear, right? The epiglottis is what covers your trachea, your respiratory system, when you swallow food, when you swallow a drink, that would protect anything from entering into your respiratory system, into your lungs. Epiglottis. And the eustachian tube. The eustachian tube would be what equalizes the pressure in your middle ear. So when you go to a higher elevation and you hear popping noises, that's your eustachian tube opening and closing and able to equalizing the pressure in your middle ear. So know that those are very important to realize, that the E's for location for elastic cartilage. Fibrocartilage, think your intervertebral discs, the disc between each of between the vertebra of the spine. Think your pubic symphysis, and I'll show you this in just a moment. Think also your meniscus of your knee and the TMJ. So, right, you're able to open and close your jaw. Well, that's the temporomandibular joint. And even if you take your fingers, number 2 finger, right, put them in your ears, open and close your jaw. Right, you can feel, you can palpate the temporomandibular joint. There's a piece of fibrocartilage there in order to allow for a lot of wear and tear. Fibrocartilage is very strong cartilage. Okay? Let's take a look here. So as we're looking at this image, and I'm going to post this image for you all, okay? This image here shows you that in red, fibrocartilage, the pubic symphysis, fibrocartilage. You're not seeing the TMJ here, but you're seeing also the meniscus of the knee, fibrocartilage. The external ear, and then the epiglottis, right? Eustachian tube you're not viewing, elastic cartilage. All else is light blue. That would be considered hyaline cartilage, okay? Now, hyaline cartilage, very unique in its presentation in that, so see, this is a good image of hyaline cartilage that you would see in the lab. So you see here very easily that you're not seeing a whole lot of fibers in comparison, right? And you're seeing here that we have these cartilage cells, these chondrocytes, present within a space called the lacunae, right? Or lacunae plural, lacuna singular. And so they're spaced out throughout this matrix of the hyaline cartilage. All the other areas of the body that I didn't mention to you as far as for elastic and fibro will be hyaline cartilage. So your nose, hyaline cartilage, right? Your costal cartilage right here, hyaline cartilage. The ends of bone, hyaline cartilage, right? That's called articulating or articular cartilage. Protects the ends of the bone of joints. Know that the hyaline cartilage model, that when a fetus is developing within the womb, right? And this child, as it produces its skeleton, starts off as hyaline cartilage and then ossifies to become bone tissue in your skeleton, your hard skeleton. Okay. So all the other areas of the body, hyaline cartilage. So those would be then as far as connective tissue. We're going to look at bone and blood next, as far as finishing out our connective tissue. So let me close these tabs, and then we'll begin with. Okay, so let's look at compact bone, and then we'll look at spongy bone. All right, now before we do that, let's take a look at an image, and just to show you, this is a good. Okay, so as we take a cross section of a bone, okay, we'll see that there is compact bone on the outer portion and spongy bone on the inner portion of the bone. Okay, let's look at this image right here. Compact bone, spongy bone. Spongy bone has holes present, right? So let's look over here. Take care, son. Spongy bone, compact bone. As we're looking at the end of a bone, this is spongy bone, where we're going to have red marrow present in order to produce blood cells, and then yellow marrow present here in the medullary cavity. This yellow marrow is adipose tissue. It's fat storage. Okay, so we have this two different types of tissue, and what's taking place with them. Go to our PowerPoint for a moment here. All right, so what you're seeing here with this slide is we're looking at compact bone. This is an example of compact bone. You'll see here, this structure right here is a cylindrical type structure. So the shaft of a bone, so if we're looking at too long bone here, or the long bone here of the humerus, right? So long bone, shaft of the bone is called the diaphysis, okay? You'll have these tubes of bone, these osteon. So I thought I had, I don't have it here. My roll of paper towels, sorry. But when you're thinking of a roll of paper towel, it's very similar to what you're thinking of the osteon, in that there are, instead of layers of paper towel, we have layers of bone, and in the middle, right here and right here, it's a hole. And this hole is present for blood vessels, nervous tissue, nerves, also lymphatic vessels. That's present within the hole right here, the central canal of the osteon, of this layers of bone, okay? That's the compact bone. Let's see here. It's not giving me, okay. So let's minimize this. So we're gonna come back to this image here. This is good. We're seeing here that the end of the bone, this is spongy bone, and here we would have lining the medullary cavity would also be spongy bone, and the outer part of the bone is the compact bone. Compact bone, spongy bone, and bone is covered in what's called periosteum. It's this connective tissue covering of the outer portion of bone. But here we have this compact bone, the spongy bone. The spongy bone is made up of, comprised of these rods of bone. See how there's openings, rods of bone. Compact bone, very compact, very tight. But know that it's cells present in a matrix. Unlike epithelial tissue, which is cells that are very close together, right? Tightly packed cells. Not in the case of connective tissue. Yes, there's quite a few cells here, but they're still present within this matrix of these layers of bone. Okay? And so these osteocytes are present within lacunae also. Okay? These lacuna would be singular, lacunae plural, for these little spaces within the compact bone, between the layers of the bone. So let's now go back to, let's see here, compact bone histology. All right. So here we would have, so this would be something that would very closely approximate what you'd see in the lab, which would be, here's an osteon. It's this cylindrical, concentric layers of bone, right? And what happens is that we have these central canals for the blood vessels and different other vasculature. And know that you'll see that we have these osteons present throughout, and they're in different stages as far as some of them are more complete as far as you're seeing this whole cylinder, and some are not, because bone is continually being replaced or remodeled. Take a look at this image here. Again, it looks almost like a tree trunk was cut, and you're seeing the tree trunk itself that's left. That's the osteons right here. And the hole in the middle is the central canal for the blood vessels. Okay? And so compact bone is very unique in how it looks. Here we go with another image. Here's the osteon, layers of bone, and this haversian canal, it's an old term that's used for the central canal. Lacuna, it's a space where osteocytes are present within. And when you're looking at this image here, see all those little black dots? Those are lacuna, or lacunae, plural, and an osteocyte would be present in those spaces. Compact bone. Now, spongy bone, again, spongy bone histology is going to have a different appearance. Now, this one, I'm just showing you kind of, I'm stepping back a bit and just showing you that, see all these pink lines? These are the trabeculae. These are the rods of bone in spongy bone. The areas that are lighter colored, this would be the bone marrow present in spongy bone. So take a look at this image here. This would be spongy bone, and here would be marrow, bone marrow, okay? So just so that you can see the difference between, so again, we're looking at here, here's the trabeculae, and in between would be the bone marrow. And in particular, wow, doesn't that look like adipose tissue? It looks like those fat cells that are present. So this would be more in the medullary cavity and not at the ends of the bone. Okay, so compact and spongy bone. And then lastly, we're going to look at the last type of connective tissue, and it's a fluid connective tissue, a blood smear. And as we're looking at a blood smear, it's very, very, this is another one that's pretty easy to spot. And what you're looking at right here, folks, is that we have many red blood cells, and here's a white blood cell right here. Here we have, in this image, many red blood cells. Here are the white blood cells stained purple. And I'm not really seeing, oh, there we go. There's a platelet right there. That little dot right there, that's a platelet. Let's take a look at another image. So here we have the little platelets, white blood cells, red blood cells predominant throughout, okay? Platelet, white blood cells, red blood cells predominant throughout this example of a blood smear, okay? So this is the last, that's called a fluid connective tissue. These are the connective tissues of the body. I'd like to show you now. We're going to look at skeletal muscle, cardiac muscle, and smooth muscle, okay? So here we have skeletal muscle, and let's take a moment and go back to our PowerPoint just for a moment here, chapter 5, and we'll look at, so here in this image, this is showing you first smooth muscle. And smooth muscle, you can see here, it's like these spindle-shaped fibers, and there's no lines here that would show you, like it looks like there are, we call them striations. that's not present in smooth muscle. And what you're seeing when you're looking at this smooth muscle, it looks like the nuclei have been stretched a bit. They've been taken and stretched a bit. Smooth muscle, these spindle-shaped fibers. Skeletal muscle, look at how different that looks. See these striations? They're present there, okay? And you see the fibers one on top of the other. Skeletal muscle, voluntary control. Cardiac muscle, there's branching of the fibers, the cardiac muscle fibers, and there are these darker lines. There are lines present, but there are also darker lines also. These darker lines are called intercalated discs, and they allow for communication and contraction of the musculature. So when we think of the cardiac muscle, it's the chambers of the heart. So the upper chambers are the atria, the lower chambers are the ventricles. So the atria contract as one, the ventricles contract as one. And as a result of these intercalated discs, it allows for communication between these cardiac muscle cells. Okay, so let's now go to the internet and to Google and see as far as skeletal muscle histology. So this is a good image right here. You can see these striations. This is voluntary muscle, long muscle fibers. So know this, folks, that your biceps brachii muscle. So the muscle belly from the beginning, from really the start to the end, the proximal to distal portions of the muscle belly, right? That muscle belly, those fibers, or the skeletal muscle fibers, go from here to here. They go from the length of the muscle, from tendon to tendon. That's these muscle fibers. Okay? Very unique in their appearance. Here's another image. Right? You see the striations? Skeletal muscle fibers. You see that on the periphery, you have these nuclei present. Oh, look at this image. This is good. So let me just this image here. Good. Get bigger. So here's just, this is just nice, and that's showing you have the different types of tissues right next to each other. Skeletal muscle, cardiac. See here these intercalated discs. And smooth muscle. It looks like the nuclei have been stretched in comparison to the others, and they're centrally located in the muscle, not and centrally located here for cardiac muscle, but on the periphery for skeletal muscle. I'll share this image with you all, and you can enhance it. Okay. Again, when I'm testing in the lab, I like to do the representation of seeing that, hey, you can see that these nuclei look like they've been stretched, and that's a really good issue that you're thinking regarding smooth muscle. Okay, here's another one example. See those nuclei? They look like they've been stretched. Simple, smooth muscle. Sorry, not simple. Smooth muscle. Cardiac muscle. Yeah. So let's give you an example here. And what you're seeing is here, there's examples of, you can see it's striated, see the striations, but then you can also see some dark lines here and there. Those are those intercalated discs, see right there? That's helping you, and also see some branching going on. That's cardiac muscle. Here we go, another example. See those dark lines? See, it's all striated, but you can also see these dark lines. Those are those intercalated discs of cardiac muscle. And you can see branching on both of those images. And here again, looks like it's striated. They've also got darker areas for these intercalated discs, and that's cardiac muscle. And lastly, we'll look at the neuron smear. And so folks, you need to, I said, look at many, many images in order to determine what your be able to determine what you're looking at when you're seeing these in the microscope and in the lab. So take a look at this right here. What we're looking at is multipolar neurons, multiple processes of these neurons. This is a soma, the neuron cell body, and we have these different processes that are projecting off of or projecting into the neuron cell body. Here we can see they've identified, as far as here's the axon, information will be going away from the neuron cell body, and then these dendrites will bring information to the neuron cell body, and there's the nucleus region of the neuron cell body. The soma is a term that we'll use. Take a look at this image here. You're seeing multiple, these are multiple multipolar neurons. Multiple multipolar neurons. And you're seeing these little dots surrounding, those are the neuroglial cells. Those are the support cells for the neuron. Protection, they give nutrition, they really just help the neurons do their job. Okay? Neuron smear. Multipolar neurons. Okay, folks, so that's for the histology. It's intense and there's a lot of information to review and go over, but you can do it. You can do it. And I'm going to now go into chapter 3 and finish up chapter 3 for you all. Okay. So in chapter 3, I would like to start with this slide here and then go to the end of the chapter. And so what we're looking at here, folks, is that we've talked about movement from outside to in and inside to out of the cell through the plasma membrane. Recall that this plasma membrane is a phospholipid bilayer. It's a fat, right? It's comprised primarily of fat, but there's also proteins, cholesterol present interspersed throughout this plasma membrane, which helps to encompass the cell and keep all the guts of the cell inside and be selectively permeable, meaning that some substances can enter in or leave, but not all. And they might need channels in order to do this. And these channels are proteins. So here we have a leak channel, and this leak channel will allow for movement of material. In particular, we're looking at sodium ions, and these sodium ions will move from their area of high concentration to the area of low concentration. That's diffusion. It's a passive process, right? Movement of high concentration to low concentration. Going from high to low, diffusion. And if we have this channel, now this channel is open, leak channel, it's always open, right? This will allow for this to take place. Now, we'll need energy, ATP, energy to be released in order to allow for substances to go from their low concentration to their high concentration. This is against what we would say is the concentration gradient, right? And you need that. So in order to go from low to high, it's opposite of the passive, and you need energy in order to do that. And we move 3 out and 2 in. 2 potassium in, 3 out as a result of this sodium potassium pump that requires ATP in order to do this. So this is a concept you need to really master and understand, that concentration gradient, we're moving from high to low, that's diffusion. Passive process. If we're moving from low to high in the opposite direction, then you're going to need energy in order to do that, okay? Active process. So passive process, diffusion from high concentration, the brackets represent concentration, to low concentration. So here we have high concentration, low concentration. We're moving through the plasma membrane from outside of the cell to inside of the cell. And so that movement is diffusion from high to low. Here is representative of equilibrium. Equal concentration here as well as here, outside and inside, okay? Osmosis is the movement of a solvent. So here we have solutes moving. The blue dots, solute, okay? That's moving through the plasma membrane. And the white would represent the solvent. Many times it's water, okay? Well, in the case of osmosis, It's diffusion of the solvent, water, across a semi-permeable membrane, where the solutes are not going to be allowed to move, but the solvent can. So here we go. As far as this is diffusion of water, aka osmosis, and this is where we have high concentration of water here, or solvent, right? Low concentration of solvent here. High concentration of solute, low concentration of solute. But we're thinking high to low. So high concentration of solvent, the water, will move from high to low. So that's what it does. And now we have equal concentration here as well as here. Not the equal quantity, but the equal concentration between solute and solvent. The solvent is pink in this demonstration here. So tonicity. Tonicity, we're thinking of what's taking place within the liquid portion of the blood, the plasma, and how it can have an effect on the red blood cells that are circulating throughout. And really, the red blood cells are the primary cells present within the blood, okay, and the highest concentration. Isotonic conditions are conditions where there's a slightly salty environment. So think of saline solution. Saline solution is an isotonic solution, and saline solution has a little bit of a salt present, not a whole lot though, slightly salty. Red blood cells are not affected by an isotonic solution. So the plasma, when it's isotonic, water will move in and out of the red blood cell, but will not damage the red blood cell. But in the case of a very salty solution, or so the opposite of the salty solution would be minimal, if any, salt solution present, a hypotonic solution. So in the case of drinking salt water when you're out at sea and you have no other water, without doing anything to remove the salt, you'll die. You'll die a slow, painful death. Your red blood cells will shrink, they'll crenate, they'll die. In the case of a hypotonic environment, if you drank a couple gallons of water very quickly, all in a row, in a very short period of time, you can create a hypotonic environment, and this will also be detrimental to the red blood cells, leading to then swelling and lyse, then expanding and bursting. So here we have these examples of a very salty environment, or the opposite of that salty environment, a hypotonic solution, and they'll swell, the water will enter into the cells, the red blood cells, and they'll burst, or the water will exit from the red blood cells and they'll shrink and they'll crenate. Both instances, the cells die, they're not carrying oxygen to the tissues. This can be deadly to the human body. Isotonic situation, we have waters moving in and out, not damaging the red blood cells. They're able to do their job. In the case of active transport, so I mentioned to you the passive transport, where we're having movement taking place, no ATP is required, no energy is required. What active transport, this is important. We need ATP to be present. Now, we can have different carrier proteins, and in the case of the sodium-potassium pump, this is what I showed you an example of before, this antiporter. This is where we have sodium. Let's go back here for a moment. I want to show you this. So sodium is moving out, 3 sodium are moving out, 2 potassium are moving in, energy is required. So, and it's just on the opposite side, but 3 sodium are moving out, 2 potassium are moving in against their concentration gradient. So this would be then an active process because ATP is required. And you'll see here as far as uni and synporter, what's going on with those. So again, the sodium-potassium pump and antiporter, 3 potassium ions are going out, 2 potassium ions are coming in. Against their concentration gradient, we need ATP in order to do this. I mentioned to you regarding the leak channels already. They're always open, and material are moving, ions are moving from high concentration to low concentration. They're diffusing through these leak channels. They're always open. In the case of gated channels, we're going to see this as we study throughout this semester and next semester. You'll either have, and so a gate, think of a gate as having, you need a key in order to open the gate, okay? So the key can be for these gated channels, the chemical, small molecule, like this neurotransmitter, acetylcholine. And this would be for the, at the neuromuscular junction where muscles are able to contract. ligand-gated channels are located there. In the case of a voltage-gated channel, it's an actual change in the voltage and change of the charge will be the key to open the gate. So see here we have different representations of channels and what's passing through them. And you'll see here the representation of a gated channel. Excuse me, one moment. In the case of a chemically gated channel, this ligand-gated channel, we'll have acetylcholine. It's a neurotransmitter present within the nervous system, and this will allow for it to interact with specific receptors that will in turn act as the key to open this channel. In the case of voltage-gated channels, here it's closed, here it's open. And as a result of a change in the polarity here in the voltage, that will cause that to open. Now, you will learn more about this voltage and what's going on with this. We're going to say the term resting membrane potential. We'll talk about that as the semester goes on, but just introducing it to you right now. So you see here these phospholipids, bilayer, plasma membrane. Now, a mechanically gated channel that responds to, mechanically gated, so it's applying to pressure, will act as the key to open this channel and allow for ions to pass through. This term G protein complex is very important to know that and to recall that it allows for changes to take place within the cell when a substance cannot enter the cell. So a substance that can't pass through the plasma membrane, but it still needs to cause some type of change to take place within the cell, the G protein complex will allow for this. And so an example would be water-soluble hormones cannot pass through the lipid plasma membrane. So they'll interact with these receptors outside of the cell, outside on the plasma membrane, which will cause change to take place within the cell. So look at this here. This is just an example to show you. So here's this chemical, this molecule, that will interact with a specific receptor for this molecule, which will in turn cause change to take place inside of the cell. Again, another example of this. Here's this water-soluble hormone. Can't pass through the plasma membrane, but it can interact with this receptor, specific receptor, which will in turn cause changes to take place inside of the cell. Now, know that there is substances that will enter into the cell, substances that will exit out of the cell. When we have substances entering into the cell, EN, entering, endocytosis. Whether we're taking in liquid material or solid material, pino and phagocytosis, or whether we're actually taking in substances that are connected to receptors that will then bring them inside of the cell. This is endocytosis, substances entering the cell. Exocytosis, what do we see here? EX, exiting, leaving the cell. The substances that are leaving the cell, so substances that we can actually be produced by the cells would be enzymes, proteins, mucus, another type of protein, water, and breast milk, secretions and such. These can leave the cells via exocytosis, exiting the cell.